Hello, my name is Thorsten Balke and I'm a senior lecturer at the University of Glasgow. At this year's MASTS Annual Science Meeting, I will be speaking about mangrove habitats and their restoration, and I will highlight some of the research we are currently doing with partners in Southeast Asia. If you'd like to know more, please join the presentation.
At this year's annual science meeting, I'll be presenting a mathematical model which aims to understand the spread of the invasive seaweed Sargassum muticum in a theoretical framework. This study is part of my PhD research, which focuses on modeling the dynamics of this alien species in Scottish waters. Investigations show that in the years to come, climate change will increase the spread of this highly invasive seaweed in Scotland and the UK, and consequently its negative impacts. Here I will walk you through the steps of building a model that incorporates the key life history stages of this macroalga. It is characterized by floating fragments that detach from the adult plant and are able to release spores as they drift for long distances. These appear to be critical in the invasion process, but can hardly be quantified, which makes modeling an interesting tool to use for this problem.
Hi, I'm Juliet, a postgraduate applied marine science student from Plymouth University, and I'll be talking about sediment stability in intertidal habitats at the MASTS Annual Science Meeting in October.
I am going to talk about self-regulation processes in kelp forests. To understand how a forest is sustained, we need to determine what regulates the density, growth, and recruitment of the individual kelps. Some investigators have drawn parallels between marine and terrestrial forests, with competition for light producing a strong density-dependent response in the recruitment of young individuals and in the growth of established plants. In kelp farms as well, denser stands tend to lead to smaller plants with lower biomass yields. Despite the empirical evidence for strong competitive interaction between individuals, leading to the regulation at forest scale, there are very few models of kelp which adequately incorporate this process. We develop an individual-based modeling approach which resolves the age-size trajectories of competing kelp plants over time in a forest. Initial results from the model show variation in individual growth trajectories between successive annual cohorts to be strongly influenced by the density of the forest.
Hi everyone, I'm a PhD student and my research focuses on investigating primary production changes after offshore renewable energy deployments. To achieve this, comparison of chlorophyll A before and after these deployments can be made using satellite information, which still have problems with subsurface representation. We found out that in situ chlorophyll A is related to satellite data up to 12 meters depth, where the effect of the depth on satellite sampling starts to be negative. We compared in situ characteristics to satellite data, and almost 90% of our samples show the depth of the maximum concentration below the capacity of satellite sampling, suggesting possible underestimating issues of chlorophyll A using satellite data. To correct this bias, we developed a model to relate subsurface characteristics to surface physical variables, whose preliminary results will be presented in the talk.
A long-term shoreline monitoring program by Orkney Islands Council was initially aimed at assessing impacts from oil and gas-related activities. Assessment of the long-term monitoring data, with no major incident taking place over the study time, have provided a unique opportunity to establish macroinvertebrate baselines and to determine the noise within the baseline conditions. The results showed annual variation in both the composition of taxa and in the characterizing taxa identified by SIMPE analysis. Even with this annual variation present, the dominant taxa remained the same over time. These results provide a comprehensive description of baseline conditions, demonstrating the importance of long-term annual monitoring of sandy shore sites to fully understand the extent of macroinvertebrate community variability over time.